IB Bio Topic 11 Part 10, Sexual Reproduction B, will focus on female anatomy and physiology, as well as the processes related to oogenesis. The essential idea is sexual reproduction involves the development and fusion of haploid gametes. Here is the outline of all the available movies for Topic 11, Animal Physiology. Use this outline to find the movie you need for review. This movie is focused here. This image nicely shows the position of the paired ovaries in the body, the uterus, and the vagina. Here are two IB syllabus statements that should be review for you. Annotate diagrams of the adult male and female reproductive systems to show the names of structures and their functions. Outline the role of the seminiferous tubules, epididymis, seminal vesicle, and prostate gland in the production of semen. In the same way that the male reproductive system has paired testes, the female reproductive system has paired ovaries. One here, one here. The oviduct connects the ovary to the uterus. The soft tissue lining the uterus is known as the endometrium. The uterus opens to the vagina through the cervix. Sperm must move from the vagina through the cervix into the uterus and then into the oviduct to reach an egg that's been released from the ovary. One last note, follicle is the structure within the ovary that develops and then releases an egg into the oviduct once per month. Can you identify the structures and their functions? I've color-coded in blue the structures directly related to reproduction. One is the ovary. Meiosis occurs here to produce and release an egg. Two is the oviduct. The oviduct moves the egg from the ovary to the uterus. Three is the uterus. Development of the embryo occurs along the wall of the uterus. Four is the cervix. At birth, the child is pushed through the cervix by muscles along the wall of the uterus. Five is the vagina. While the baby is pushed through the vagina at birth, the vagina also serves as the receptacle for sperm. Now, six is the bladder. It holds the urine for eventual release. And seven is the urethra. Urine is released through the urethra. Here's an image of one ovary with its adjacent oviduct. The egg is released into an abdominal space that lies between the ovary and the oviduct. The fimbriae and cilia serve to move the egg into the oviduct toward the uterus. Oogenesis in females involves a complex interplay of hormones to release one egg per month on a cycle known as the menstrual cycle. In the course of the month, the anterior pituitary releases FSH and LH. The ovary releases estrogen and progesterone. And within each menstrual cycle, a single ovum, egg, develops and is released, and the uterus readies for implantation of an embryo. The details of the hormones and the events of the menstrual cycle were presented in Topic 6. If you have forgotten the details of the menstrual cycle, you need to go back and review. FSH from the anterior pituitary targets the ovary, initiating the development of a follicle seen here, within which is the developing egg. A surge of LH from the anterior pituitary results in ovulation, the release of an egg, but the secondary oocyte has only proceeded through meiosis 1, not meiosis 2 yet. As well, during the 28-day menstrual cycle, the endometrial lining of the uterus, seen here, is developing, thickening, readying for the implantation of an embryo. This structure here is known as a blastocyst. This will develop into an embryo. Now, this image shows sperm arriving in the oviduct, resulting in fertilization. But if fertilization does not occur, which is most often the case in women, the egg and the endometrial lining are flushed out of the uterus in a process known as menstruation, sometimes called a period. Here is the first IB syllabus statement that forms the basis of this movie. Outline the processes involved in oogenesis within the ovary, including mitosis, cell growth, the two divisions of meiosis, the unequal division of cytoplasm, and the degeneration of the polar body. A cell within the ovary, the diploid oogonium, undergoes mitosis to form two cells, only one of which proceeds into meiosis 1. Of the two cells formed following mitosis, the one that enters meiosis is known as the primary oocyte. The primary oocyte is diploid. Women are born with approximately 300,000 primary oocytes arrested in prophase 1 of meiosis. At puberty, the menstrual cycle is initiated with the development of one primary oocyte into a haploid secondary oocyte every month. Keep in mind that meiotic divisions are unequal. One cell holds more cytoplasm than the other, and a polar body is formed and it degenerates. The secondary oocyte is released from the ovary at ovulation, arrested in metaphase 2. Unless fertilization occurs, the secondary oocyte never completes meiosis 2. Here is a diagram of the ovary showing the primary follicle, within which is the diploid primary oocyte. Each month, FSH, hormone, prompts the primary oocyte to go through meiosis 1, becoming a secondary oocyte within a larger structure known as the follicle. To clarify the terms here, the follicle is a multicellular structure composed of an oocyte and many other cells, some of which are closely associated with the egg, providing nutritive support. But some of the cells of the follicle are endocrine in function, releasing estrogen early in the 28-day menstrual cycle, and progesterone in the second half of the cycle. The estrogen targets the lining of the uterus to stimulate the development of the endometrium in case fertilization occurs. Keep in mind that the follicle is a large multicellular structure that includes the egg, nutritive cells, and cells with endocrine function. Once again, the primary follicle develops under the influence of FSH into a secondary follicle. Meiosis 1 has occurred. The mature secondary follicle ruptures. This is known as ovulation, releasing the secondary oocyte into the oviduct approximately on day 14 of the menstrual cycle. The part of the follicle that remains within the ovary becomes what's known as the corpus luteum. The corpus luteum continues to secrete progesterone and estrogen in the second half of the menstrual cycle. These hormones target the endometrium of the uterus to ready the lining of the uterus for implantation of an embryo in case of fertilization. Let's bring this IB syllabus statement into play because it coincides with everything that I'm covering here. Annotate a diagram of the ovary to show the stages of gametogenesis, including germinal epithelium, primary follicles, mature follicle, and secondary oocyte. The primary follicle develops under the influence of FSH into a secondary follicle. Meiosis 1 has occurred from the primary follicle to the secondary follicle. The developing follicle is also secreting estrogen. The follicle is a large multicellular structure with a developing egg, nutritive cells, and endocrine cells. The mature follicle, with its fluid-filled chamber, ruptures, releasing the secondary oocyte into the oviduct approximately on day 14 of the menstrual cycle. This is known as ovulation. The secondary oocyte, the egg, remains surrounded by many attendant cells. The ring of cells surrounding the egg is called the corona radiata. The part of the follicle that remains within the ovary becomes what's known as the corpus luteum. The corpus luteum continues to secrete progesterone and estrogen in the second half of the menstrual cycle. These hormones target the endometrium of the uterus to ready the lining for implantation of an embryo in case of fertilization. Here's another image of the ovary and the development of the follicle. FSH initiates the development of the primary follicle to secondary follicle. Meiosis 1 has occurred from primary oocyte to secondary oocyte. Ovulation of the secondary oocyte can be seen here, and the corpus luteum remains within the ovary as an endocrine structure. Remember that the secondary oocyte has completed meiosis 1, but not meiosis 2. Here is a micrograph of the mature follicle just before ovulation within the ovary. The follicle is a multicellular structure with a fluid-filled chamber and follicle cells surrounding the secondary oocyte annotated in red. Here is a photograph of ovulation. The wall of the ovary bursts, releasing the secondary oocyte somewhere in the middle of this mass of cells. All of these cells are attendant and will follow the secondary oocyte down the oviduct. Let's review. A cell within the ovary, the diploid oogonium, undergoes mitosis to form two cells, only one of which proceeds into meiosis I. Of the two cells formed following mitosis, the one that enters meiosis is known as the primary oocyte. The primary oocyte is diploid. Each month, a primary oocyte is stimulated to undergo meiosis I to form a haploid secondary oocyte and one polar body. Keep in mind that the meiotic divisions are unequal. One cell holds more cytoplasm than the other, and the polar body degenerates. The secondary oocyte is released from the ovary at ovulation, arrested in metaphase 2 of meiosis II. Unless fertilization occurs, the secondary oocyte never completes meiosis. Here's the next IB syllabus statement. Annotate a diagram of the mature egg to indicate functions. The secondary oocyte is a complex structure that has resulted from meiosis I. Thus, there is a polar body present just outside the plasma membrane. Also lying just outside the plasma membrane is a glycoprotein layer known as the zona pellucida. Sperm will need to penetrate the zona pellucida in order to achieve fertilization. The internal structures include many structures familiar to you and some new. The cortical granules are Golgi vesicles modified to perform a function known as the cortical reaction. More on this later in the movie. Remember, the egg is large and contains proteins and nutrition such as lipid droplets that provides for the mitotic development of the embryo just after fertilization. Microvilli increase the surface area of the plasma membrane to facilitate the absorption of added nutrients from the follicular cells that surround the secondary oocyte. Here is an image that shows the mature follicle here on the left just before ovulation and the secondary oocyte just after ovulation. The cells seen here on the mature follicle become the attendant cells, nutritive cells, to the secondary oocyte following ovulation. You can see the glycoprotein layer known as the zona pellucida here, and the mature egg cell lies within. Here is a list of the unique aspects of secondary oocyte structure, some of which should be included in your annotation of a drawing of the egg. Receptors for sperm cells, nutrients for development such as lipid droplets, stored mRNA for development, stored protein for the early divisions, the mitotic divisions after fertilization, the protective coat, the zona pellucida, and cortical granules. More on these structures later in the movie. You can see the secondary oocyte released into the oviduct by the process known as ovulation. The secondary oocyte is surrounded by attendant follicle cells. In this image, we can see the secondary oocyte. Here is the plasma membrane of the secondary oocyte. Here is the zona pellucida. And here's the polar body resulting from meiosis I. As well, we can see fertilization. The sperm have reached the egg in the oviduct and penetrated the follicle cells, penetrated the zona pellucida, and reached the plasma membrane. Upon delivery of the haploid sperm nucleus, the secondary oocyte undergoes meiosis II, as you can see. Once again, ovulation around day 14 of the menstrual cycle. Cilia move the secondary oocyte down the oviduct. You can see follicle cells and the zona pellucida. Sperm arrive to achieve fertilization. Fertilization is the joining of two haploid nuclei. The diploid zygote undergoes a series of mitotic divisions to become a multicellular structure known as a blastocyst. And the blastocyst implants in the uterine lining, being supported by progesterone and estrogen secreted by the corpus luteum. Remember, the LH surge results in ovulation around day 14 of the menstrual cycle. The secondary oocyte is released into the oviduct. Following intercourse, sperm would fertilize the egg in the oviduct, as can be seen here. I will cover details of fertilization later in this movie. Development of the embryo to a blastocyst will be covered in the next movie, part 11. Don't forget that the corpus luteum secretes progesterone and estrogen that promotes the development of the endometrial lining of the uterus in case fertilization occurs, resulting in an embryo. This slide should be review from topic 6, but I will add some new details, so listen carefully. You can see the LH surge here. Ovulation occurs around day 14, and the development of the endometrial lining under the influence of estrogen and progesterone being secreted by the corpus luteum. Now, if pregnancy does not occur, the corpus luteum degenerates, and estrogen progesterone levels go down, and the endometrial lining is shed, which is day 1 of the next cycle. But if fertilization does occur, as is the focus of this movie, the developing embryo implanted in the uterine wall secretes a hormone called HCG. This is new, human chorionic gonadotropin, HCG, and this maintains the corpus luteum for the first 3 months of pregnancy. The HCG being secreted by the developing embryo sustains an active corpus luteum for the first 3 months of pregnancy. I will cover HCG in greater detail later in this movie and in the next movie, part 11. Off-the-shelf pregnancy kits test urine for the presence of HCG using monoclonal antibodies. Make connections, ask questions. Again, this slide should be review from topic 6. Under the influence of FSH, the primary oocyte develops into a secondary oocyte. Under the influence of LH, the secondary oocyte is released into the oviduct on approximately day 14 of the average 28-day menstrual cycle. The endometrium of the uterus develops under the influence of estrogen and then progesterone. The remaining follicle within the ovary, known as the corpus luteum, secretes progesterone to maintain the endometrium of the uterus as the uterus readies for an embryo. Here is a micrograph of a sperm cell penetrating the zona pellucida to reach the plasma membrane of the secondary oocyte. With this slide, let me transition to look more closely at fertilization. In order to achieve internal fertilization, the male sperm need to be released within the vagina of the female. You can see the penis here within the vagina. This is known as intercourse, and the sperm are released here near the cervix. Remember, fertilization in animals can be internal or external. Here is a micrograph of the egg surrounded by many sperm. Notice the size difference. External fertilization can be seen here. Review the details that I've presented on this in Part 9, Reproduction A. Internal fertilization can be seen here. Review the details that I've presented on this in Part 9, Reproduction A. Remember that fertilization occurs in the oviduct, and notice all the sperm that have arrived at the secondary oocyte. How is it that the egg is fertilized by just one sperm? How is polyspermy prevented? So here are the next IB syllabus statements. Describe the process of fertilization and the mechanisms that prevent polyspermy. Fertilization involves the acrosome reaction, the penetration of the egg membrane by a sperm, and the cortical reaction. In this image, taken as a time sequence left to right, 1, 2, 3, 4, we can see sperm arriving just outside the zona pellucida. The acrosome, a vacuole of enzymes within the sperm cell, breaks open to digest away the zona pellucida. Once the sperm cell membrane fuses with the egg plasma membrane, seen here, cortical granules within the egg exocytose their molecules just outside the plasma membrane of the egg. The cortical reaction results in the sloughing off of the zona pellucida. The zona pellucida peels away, removing any other sperm that might be competing with this sperm for fertilization. Once again, left to right, we see the arriving sperm passing through follicle cells to reach the zona pellucida. The acrosome breaks open, releasing enzymes that digest through the zona pellucida, allowing the sperm membrane to contact the egg plasma membrane, fusing the two cells. Cortical granules within the egg release their contents outside the egg cell through exocytosis, causing the zona pellucida to peel off. The materials within the cortical granules are hypertonic, which draws water into the space between the egg membrane and the zona pellucida. This pushes the zona pellucida off the egg, taking any competing sperm with it. In this micrograph, we can see two nuclei joining, one from the sperm, one from the egg. This is fertilization. At this point, the oocyte has fully completed meiosis II. The polar body of the second meiotic event is seen here. The cell is diploid, known as a zygote, and it is ready for mitosis as the embryo grows. Here is the sequence of events leading up to fertilization and the formation of the diploid zygote. I've circled the events that require some explanation, including appropriate language use. Successful fertilization requires only one sperm fusing with the egg. To prevent more than one sperm reaching the egg at the same time, known as polyspermy, there are two mechanisms to prevent polyspermy. First, there's the fast block. The fast block is an electric-like event that changes the charge surrounding the egg plasma membrane. Sodium channels open, sodium diffuses in, the membrane depolarizes, and polyspermy is blocked. This should be somewhat familiar to you from our study of neurons, but you actually don't need to know fast block for our study of reproduction. But the cortical reaction, the second reaction, is more slow than the fast block, and in this case, cortical granules exocytose materials into the space under the zona pellucida. This causes the zona pellucida to peel away. There are enzymatic events, there are osmotic events, but polyspermy is blocked. Once fertilization happens in the oviduct, the diploid zygote rapidly undergoes mitosis to become a blastocyst that implants in the uterine wall. With pregnancy, the embryo secretes HCG that targets the ovary, specifically the corpus luteum. HCG, as a hormone secreted by the embryo, sustains the corpus luteum for the first 3 months of pregnancy. The corpus luteum continues to secrete progesterone and estrogen to maintain the endometrium where the embryo is growing into a fetus. Here is the relevant IB syllabus statement, outline the role of HCG, human chorionic gonadotropin, in early pregnancy. We can see the fetus in the uterus here, and the developing embryo secretes HCG that targets the ovary to maintain the activity of the corpus luteum. The corpus luteum continues to secrete estrogen and progesterone to maintain the endometrial lining of the uterus. After the first trimester, 3 months, the corpus luteum degenerates, and the placenta takes over the secretion of progesterone and estrogen to maintain the uterine lining throughout the remainder of the pregnancy. So now, if you've been listening and thinking, you can add another organ to your list of endocrine glands. The placenta is an endocrine gland, as it secretes estrogen and progesterone in the second and third trimesters of pregnancy to maintain the endometrial lining of the uterus. HCG stimulates the ovary to secrete progesterone during early pregnancy. In this graph, we can see the sharp spike in HCG following fertilization. Through the first 3 months of pregnancy, HCG secreted by tissue surrounding the embryo targets the corpus luteum in the ovary to maintain the activity of the corpus luteum. Home pregnancy kits use monoclonal antibodies to test the urine for the presence of HCG. If HCG is detected in the urine, pregnancy has occurred. Here is a table that outlines the functions of HCG, estrogen, and progesterone. This is a slide you can study on your own. I shouldn't need to review this material again. And that brings us to the end of IB Bio Topic 11, Animal Physiology, Part 10, Sexual Reproduction, B.